Our next speaker is Orit Rozenblatt-Rosen. Orit is the scientific director of the Carmen Cell Observatory and the lead scientist at the Broad Institute for the International HCA Initiative. She is the co-chair of the HCA Standards and Technology Working Group, the STWG, and today she will talk about towards generating a high-quality human cell atlas from benchmarks to application. I think she's gonna solve many of the questions that people have been already posting even before your talk, Orit.
So thank you very much for the kind introduction. I'm so excited to be part of this meeting. And I'm going to speak today on behalf of the Standards and Technology Working Group, or we call it in short STWG, of the Human Cell Atlas. So building a cell atlas really requires the generation of high quality data in multiple laboratories around the world. And we do so by continued contributions from the community over time. And as you can imagine, such data collection can pose several challenges. For example, data collection spans multiple labs and multiple techniques. Efforts can span years and many technical innovations that come around with the years, right? We have new technologies all the time. It requires careful experimental and flexible design. And the method's performance may vary substantially depending on the tissue or organ to which it is applied. So if I apply a specific method to kidney, it doesn't mean it will work as well on the brain. So in order to start addressing some of these challenges, the Human Cell Atlas established the STWG. And the STWG has several aims. So first, it aims to provide evolving guidelines and current best practices for protocols, SOP, as well as experimental design. Second, performs benchmark experiments on existing and new methods for single-cell genomics. Third, it developed guidelines for analytics and QC measures. Important aspect of the STWG is also that it disseminates and shares its experience. And lastly, it identified key areas for technical improvement. And so our general approach is to procure tissue from specific anatomical sites. And this can be different tissue types, resections, biopsy, autopsy, and organoids, for example, across different histological layers. And then we combine this with high-resolution measurements along two major branches, the cellular branch, where we profile cells and nuclei, and the spatial branch, where we profile cells and tissues. So in terms of sample collection, we suggest doing what we call a blend of different samples. So you can profile biopsies and resections from live donors. You can profile tissues from rapid autopsy, and you can also look at organoids. So we think that no source is perfect for tissue, but together they really complement each other. We think that pathology review with recording of precise anatomical location is very important for the atlas. We collect donor metadata and as much metadata as we can. And then we make sure to score the tissue for quality and integrity. So in the cellular branch, massively parallel single-cell methods allow us to profile a really large number of individual cells and nuclei. And these are among the primary technologies that are used by the HCA community. In parallel, single-cell genomic approaches for other genomic features, for example, chromatin, with single cell or single nucleus ATAC-seq or protein for using site-seq, as well as multiomics, are now used quite oftenly and have matured as well, and also scaled. So one of the major challenges when we profile cells and nuclei is that we lose the context of the tissue. We don't know where they were in the tissue, so it's really important to also use the spatial branch and perform multiplex in situ measurements of protein and RNA. And these methods are a little bit more new than the ones that I showed you before, but they are being adopted by the HCA community. And so overall, we recommend to, when you're profiling a tissue, we recommend to do anatomical and histological profiling, as well as molecular profiling and spatial profiling, and combine all of them together. An important aim for the STWG is actually to benchmark protocols. And you can think of benchmarking in several different ways. You can benchmark in the same laboratory using different technologies. You can benchmark technologies in different laboratories and use different technologies, or you can do that in different laboratories using the same technology. So these are sort of three well-known ways to benchmark. And this is something that the STWG is really invested in. And so the first benchmarking efforts were around systematic benchmarking of single cell and single nucleus RNA sequencing. And this was done by two complementary approaches. In the first one, there were a bunch of cell lines and PBMCs that were shipped across the world and profiled using different single cell and single nucleus RNA sequencing genomic methods. And in the second approach, there were 3 sample types that were actually profiled using different methods in one lab. So by doing this, we tested over 20 different single cell and single nucleus RNA sequencing method. And both studies pointed to differences in protocol performance as evaluated by different metrics such as sensitivity, throughput, and cost. And they were also tested for their power to detect genes and cell type markers. So another benchmarking effort that was ongoing was to benchmark spatial transcriptomics. And this was done on a specific brain sample by people who have expertise in spatial transcriptomics. You can see them on the left, and each one of them used their method in their lab. And then the nice thing is that they came together in what's called a Space Jam Jamboree to look at the data together and process it. So currently we have several ongoing benchmark efforts. The first one is to benchmark single cell or single nucleus HTAPP, and this is done by several different labs using several different methods. And the other ongoing effort is actually to process tissue and come up with guidelines to the HCA community about how to handle tissues. And as you can imagine, benchmarking experiments produce what we call decision trees. And these decision trees are very important in order to guide researchers to choose a protocol that's best suited to their samples and their biological questions, and also provide guidelines on how to actually optimize these types of protocols. And so I want to give you one example of how we actually generated such a decision tree, or how we go about that. And this is by actually making a single cell and single nucleus RNA sequencing toolbox. And in this case, we used it in order to profile different tumor types. You can see that we have instructions for fresh samples and for frozen samples. The fresh samples are a lot more demanding to process, and you can see that there are many more steps, while the frozen samples, there are much less steps and less involved. And so we processed 8 different tumor types with different tissue characteristics. And you can see here the fresh processing. We were very successful in processing these tumor types. However, we know that when we process fresh tissues, cell dissociation depends on the cell type and the ECM composition of each tissue. And you need to process these samples pretty quickly. And so in order to address some of these challenges, we developed quite a few years ago a method that's called single nucleus RNA sequencing that allows you to profile frozen tissues. And it has several advantages. It doesn't require cell dissociation. You can use it on frozen or lightly fixed tissues. It decouples collection from processing, which is really important. Sometimes you don't want to come late at night to process samples. Gives you access to banked samples. So if you have rare samples all around the world, you can bring them all together and process them. It allows early pooling within and across donors, and you can process tens of thousands of nuclei at once. Some of the challenges are that it's difficult to select and enrich for a specific cell type, and you have less RNA than cells. And so the first protocol for processing nuclei was used on old mouse brain. And then we decided to use that protocol on tissues from clinical samples from cancer patients. And unfortunately for us, it really did not work. And so it took us 2 years. And in these 2 years, we came up with 3 new protocols to add to our toolbox. And these are called ST buffers. There are 3 of them, NST, CST, and TST. And lo and behold, when we used these buffers, within 4 weeks, we were able to get really nice results from the same neuroblastoma tissue. And so I'm showing you here in more detail another neuroblastoma sample processed by the 4 protocols in the toolbox for nuclei processing. And what we can see is actually that we get a similar number of nuclei recovered across protocols, but we see that we get different cell type proportions with each one of these buffers. And in this case, a buffer called TST recovers the largest representation of cell types. We used this toolbox successfully across 7 different tumor types that are listed here. We can look at their proportions, and we see that overall, it seems like the TST buffer outperforms the other buffers and recovers more immune cells. And EASY, which was the first and original buffer that was used for the brain, performs the least well on most of these tissue types. Next, we wanted to compare between single cell and single nucleus RNA sequencing on the same sample. So we took a sample of neuroblastoma, we cut it in 2 and processed half by single cell RNA sequencing and half by single nucleus RNA sequencing. And you can see the results here. They seem to be similar to a poodle, that's how we call it. And what we can see is that the same cell types identified here as well, but at different proportions. And immune cells are often more prevalent in single cell RNA sequencing. You can see those here in black. And many parenchymal cells that are sort of the pink head of the poodle are more prevalent in single nucleus RNA sequencing. And so overall, we came up with guidelines of how to choose the protocol for the tissue type that you're studying. And so what we suggest is actually to compare 2 to 3 protocols in parallel on the same tissue sample. Check all your QCs, both experimental and computational. If it's possible and you have a sufficient amount of samples, compare single cell and single nucleus RNA sequencing on the same tissue sample. And there is really no such thing as the best protocol, but the best protocol for you depends on your biological question. So for example, you can choose the protocol that covers the greatest cellular diversity if you're interested in doing an atlas, right, and charting an atlas, because this gives you the most number of cells. But if you're interested in specific cell types, you can perform depletion or enrichment experiments. And so we've also used this toolbox for building atlases across 8 human tissues, which you can see here. And we really recommend using some of these tools and protocols that we provide. So one very important aim of the STWG is actually to share our knowledge and experience in working towards generating atlases. And one way to do it is by what we call a SWAT team. And this stands for short, or is described by short visits for hands-on in-person training, learning, and troubleshooting, meaning we send an experienced team of people who know how to do single-cell genomics and process samples and so forth to a place who's just learning how to do that. And together we learn what are the best practices and what would work for the other site. And here's an example of such a visit that we did as part of the Human Tumor Atlas pilot project to St. Jude's Children's Hospital. We also think that it's really important to share our protocols and our SOPs, and we do that by using protocols.io when we have a specific site for HCA where we have 156 protocols and 444 members, and there are a lot of interesting discussions about different protocols going on. So just to summarize my talk, building a human cell atlas requires flexible experimental designs, benchmarks, and best practices that can really adapt to evolving technological landscape. The HCA Standards and Technology Working Group aims to guide and advise around technologies, choices, and best practices, and disseminate knowledge. And you're always welcome to contact us. Please send us an email if you have specific questions. We combine high-resolution measurements along two major branches: a cellular branch based on profiling cells and nuclei, spatial branch to measure profiles in the tissue context. We have several benchmark projects that were performed, and the first two that I showed you are published in Nature Biotechnology, and some that are ongoing, and we're happy to share insights from all of those. We generated a single cell, single nucleus RNA sequencing toolbox to profile tumors, and this also works for additional human tissues. And we have what we call SWAT teams that provide knowledge and know-how by short visits and hands-on in-person training. And we are happy to share all our protocols and SOPs on protocols.io. And with this, I would like to really thank the Standards and Technology Working Groups, my two co-chairs, Xiaowei Zhuang and Holger Hein, and the rest of the members of the group. And thank you for your attention.
Thank you so much, Orit, for such a great presentation on benchmarking your pipelines and your protocols. It's really amazing to see all the knowledge that is already available and the discussion forums that you provide and the assistance. I think this is really amazing. And in particular for us from Latin America, we're just going to join. I think we have already all this precious information already right there available. It's really, really great. Thank you for sharing that. I do have here a question which is maybe a little bit philosophical, but taking into account all this that you've seen and all these different biological networks that you've been working with. We have a participant asking about subtypes of cells, like if the Human Cell Atlas have guidelines to determine what constitutes a cell type.
This is a very long discussion, I think. And I think people have tried to determine what a cell type is and what the cell program and what the cell state is. And I don't think that there are necessarily specific guidelines, but maybe Aviv and Sarah want to chime in on that, if they're still online.
I'm still online. I'm having some issues with my audio coming in and out unexpectedly. So I'm not sure if you can hear me now.
We can hear you.
Could you repeat that? Because I had these audio problems. I was actually working with the AV person in the background.
Yes. We have a few people here asking if there are guidelines for what the Human Cell Atlas considers a cell type.
Oh, this is the question. So first of all, no, to be perfectly honest and transparent, but it touches on a deeper question, which is, and one that you can see that people are careful about using the word type for that reason. Types get defined in different ways for different areas. And moreover, there is the overall state of the cell, which is made of many different programs, some of which represent this more invariant part of a cell's identity that we tend to call a type, whereas others are more variable and they can change for a cell of the same type. I think one of our hopes for the HCAs, I'll say two things. In areas where cell types have been defined already, and there is kind of a consensus and agreement on what they are, it is often quite easy to map that in single cell data and just see the signal. That's, the early studies in the retina were a great demonstration of that in a system where things are very stable. In tissues where work has been done now repeatedly across many groups, people are starting to converge to a consensus for the characteristics, these invariant characteristics that repeat themselves over and over and over again. And those get kind of enshrined with that terminology type. The thing that makes it complicated is these many, many other features that seem to be more variable for cells and that we tend to now call programs. And we think actually get us out of the discussion that was very complex, that was related to what's the difference between a type and a state. In a sense, any cell at any moment in time when you profile it, what you're seeing is its state. That state has features that are related to the type that it belongs to, and it has features that are related to other things, and there's relationships with those features. Yes, we would all hope that the world were a little simpler than that. I also would, the last musing I will have on this is that it really depends on who you are as a biologist in terms of what type means to you. For developmental biologists, often the type is very intimately coupled to the developmental history of the cell. For a neuroscientist, to its electrophysiological activities, and so on and so forth. And so that is one of the reasons that I think that data-driven approach can often be better than trying to just impose our pre-held conceptions.
Thank you, Orit and Aviv. That was really great answer. And we will have an insight from a developmental biologist in the next talk.